It's my pleasure to introduce Amilcare Porporato. Amilcare is professor of civil and environmental engineering at Duke University. You've been there for a little more than a decade.
Yes.
PhD from the Politecnico of Turin and stops at various esteemed academic institutions between then and 2003. Visiting chair in EPFL in 2008 and 2009. Amilcare's research covers a broad range of topics in the water and environmental sciences. His work covers hydrometeorology, ecohydrology, land-atmosphere interactions, biogeochemical cycles. I think the one unique thing that characterizes much of Amilcare's work is looking at the role of vegetation in the water cycle and the interactions between water and the biosphere. Another theme that is central to the talk is the active role of vegetation, and that's going to play a big role today, I think, in Amilcare's presentation. All yours.
Thank you, Jim. It's a pleasure to be here. Thanks for coming. I'll go right away to the talk because we want to finish within the 2 hours, as Eli said. So don't worry, no more than 2 hours. So my main research has been focusing on hydrologic variability, which is really how rainfall fluctuations propagate through soil moisture dynamics and plants, mostly to understand how plants suffer water stress. And this is really an unpredictable event for plants, and you need to study it statistically, because if you look at the rainfall time series, clearly this doesn't look like a regular analytical function. It's more like a random process. And so also, of course, with Ignacio, much of our research has been focusing on getting the probability density function of soil moisture, describing not just single trajectories, but the whole probability distribution of that. And this is the expression that we obtained analytically, and it has a very synthetic power in that it has this raw function, which is the loss function, which describes how soil and plant properties behave with respect to this key variable, which is S, which is the soil saturation. This is dry and this is wet, and of course the losses increase. And then other quantities that are key. This is again the loss function that I mentioned, but this is like the frequency of rainfall. Let's keep an eye on this because it will reappear in my penultimate slide, and I will just call it lambda and perhaps forget to redefine it. So, and then gamma, which is the mean depth of rainfall events. So this provides a synthetic expression of how likely it is that plants will suffer water stress. My research has also been devoted then to go underground in the soil and studying the carbon and nitrogen cycle in the biogeochemistry, and how soil moisture variability, which is now in this axis here, 0, dry again, and 100 is perfect, is full saturation, for example, affects microbial activity in the soil. And here is like rate of ammonification in the soil, rate of nitrification, and you see that the variability of amount of water in the soil really drives the microbial activity, which in turn affects the cycling of, for example, the nitrogen cycle between plants, soil organic matter, and ammonia and nitrate and so on. So think of hydrology being like the accelerator and the decelerator of this cycle, okay? So that's, and I've actually presented that before, including at the celebration for Ignacio a few year, 2 ago. So recently I've been much more interested in stepping back and looking at longer time scales. And the reason for that is that I've been involved in two, what I think, very interesting projects. One is the Critical Zone Observatory funded by NSF, and one of which is in Calhoun in South Carolina, and Duke University is not far from it. And this is an extremely interesting landscape, very old, very deep soils, up to 30 meters, believed to be very old, up to 5 to 6 million years old, that have been, in the words of the PI Dan Richter, soil microbiologist at Duke, has been farmed to death. This soil had been really over-farmed so that there was major erosion, major soil contamination, And this is an example of the beautiful patterns of the clays deep a few meters down. So much that now the reforestation has actually taken place, but the ecosystem has not recovered completely. And so there are interesting issues that now cover a much broader time horizon, all the way to soil formation and how soil regenerates, of course with the same processes of soil moisture dynamics and biogeochemistry. And the other interesting project is this frontier in ecosystem Earth system dynamics, which is in South America. Now this is Peru, this is in the Andes, this is the Amazon, and we have a huge gradient of elevation, lots of niches for different environmental conditions, and the idea is to understand how biodiversity came about as the geology changed and as the currents and the hydrometeorology and the ecohydrology changed. So that's the main motivation for looking at longer time scales. So what I've done recently is to basically expand my time horizon all the way to Big Bang, but now I'm 4.5 million years ago when the Earth forms, and then origin of life soon after. This is a bit quantitative. Here we have particle per million of CO2 in the atmosphere, and we start with no plants, and the first major transition for me is this one, where the first terrestrial plants came about. And this actually generated a huge drop in CO2. We will see in a moment why, not just because of photosynthesis, but because of soil formation. This is where the real soil first formed. And then, of course, these were C3 type of plants. It's a type of photosynthesis, as explained in a moment. And this is a logarithmic scale, so we're really making a huge jump now up to 30 million years ago, where you get the first evolution of major evolution in type of photosynthesis, which is called C4. Another major transition is here, around the end, here to 200 million years ago, where there were the first plants that actually controlled hydrology through stomatal control. Before it was a passive control. And finally, a little bit later on in this talk, I'll talk about the third type of photosynthesis that will appear, which is the crassulacean acid metabolism of cacti. So let's have a look at the first. This is just a little detour, and then I'll get back to plants, but more photosynthesis, I would say. But let's get to this first drop. When plants appear, what happens? Basically, there was no soil, or very little soil. And plants, they were ferns and major gigantic ferns, actually started to degrade physically the soil, and also by photosynthesis uptaking CO2 and passing it into the soil, making it, I think there is some issues with the fonts. We'll try to reconstruct that, sorry. I promise they made more sense now. Maybe we will discover new science here. But so this is just the balance equation of the CO2 in the atmosphere gets uptaken by photosynthesis. Part of this is respired back, but part of it is actually diffused into the soil. As CO2 gets there, it acidifies the soil, creates bicarbonates, which of course immediately react with the calcium carbonate, granitic rocks, and so on, and change the alkalinity. This is the balance of alkalinity, and this is the balance, used to be the balance of total carbon in the soil solution. So if you study those three equations, and the fourth one is basically the rate of change of dH dt, which is the height, the depth of the soil. And how does the depth of the soil change? Well, it changes because there is this physical weathering that introduces broken rocks into the profile, and there is dissolution, and there is biochemical weathering, and there is also erosion. So that's the balance equation that you clearly cannot see. But if you make a little bit more of assumptions, for example, that the soil solution is in equilibrium, you can actually write an interesting coupled system of two differential equations, non-linearly linked. Now, this is the rate of change of C sub A, which is the concentration of carbon dioxide in the atmosphere, which is, of course, volcanic activity plus diffusion of CO2 from the soil, plus respiration of CO2 from plants and the soils, minus photosynthesis, which is a function of biomass, which grows as the soil gets deeper. And now this is hidden because I made an assumption of steady state, and I'm just going to skip on that. But it's a reasonable assumption that the deeper the soil, the stronger and more biomass there is, and so the larger the plants. The other equation is what I described before, is the rate of change of H, soil depth, in time, which depends on how much photosynthesis there is, because I skip a few steps, but it depends on the, the photosynthesis actually is here. This is a P sub S, is not the PS that I had before. This is production, is the physical weathering. This is runoff, which drives erosion. This is mineral, this is the chemical weathering, because it has a minus in front. Sorry, I have a hard time reconstructing for a moment. And, but of course, it's proportional. This is the key thing. It's proportional to the leaching of weathered material, calcium carbonates and so on, times how much, it's controlled also by the alkalinity of the whole solution. But the key thing is that there is hydrology both in the photosynthesis, plants use water, and especially for me here, because leaching is key to remove the CO2. And once this is work done with Tony Parolari, my postdoc at Duke, and he just got these results Friday, so they're very fresh from the oven, and they're not published, but he reproduced very well the proxy data of CO2 through the Devonian here and the Carboniferous. And we were happy until we realized that we did all of this with constant leaching. And now we're speaking, of course, if I speak with Ignacio, we well know from our ecohydrological stochastic models that in reality this shouldn't happen, because leaching should decrease as plants grow, because there is more transpiration. So the depth forms, there is more storage capacity, plants can use better water to transpire, and there should be less leaching. And so this actually prompted us to come up with hopefully an explanation on that. And the first, I don't know what this is, but the first possible explanation is just simply plants facilitate now more infiltration. And so if originally the soil was not existent and there was rainfall and lots of runoff because there was not infiltration and some leaching, now because of evaporation, and the facilitation of infiltration, we have less runoff, and the difference of the two can go and maintain some leaching somewhat constant. That's a possibility. I don't like it too much because I don't think the facilitation of infiltration by plants is that important. It's probably a factor, but not that important. The explanation that I tend to favor is actually the other one, is that this was probably the first point in time in which the land-atmosphere interaction started, meaning that because of plants producing, using water and transpiring more, at that time the continent was a big Pangea, huge continent, far from most of the land was far from the sea, and precipitation was not that big probably, and having recirculation there with more evaporation, which we will see in a moment causes more precipitation, was probably a mechanism to intensify the water cycle. And by intensifying that, we can still keep decent rates of runoff, which probably were reduced a little bit by vegetation, but at the same time maintain the key hypothesis of constant leaching, which is the only way we could really justify this behavior. Because if you reduce the leaching as the stochastic soil moisture model would tell us, which independently of all the rest, is actually a reduction of this effect. And so probably it's the two effects of land-atmosphere interaction kicking in at this point, and at the same time keeping leaching fast enough to drive the CO2 depletion from the atmosphere. So that's something that is very interesting for me. I've decided to just release very recent research and maybe leave it for discussion for later. But I'll get now to the main topic of my talk, which is really plants do photosynthesis. And by doing that, they are both affected and affect the environment and the hydrologic cycle that they use. So photosynthesis, just photosynthesis 101 without offending anyone. So we are in the leaf. This is the green things here are the chloroplasts in the mesophyll, which means the middle part of the leaf. And really, photosynthesis is made of two steps. One is the light reactions, in which light is used to do photolysis, to break the water molecule and release oxygen and use protons for energy harvesting. And of course, the other part, which is really where the energy that then is transformed into ATP is used to capture CO2 and create sugars, and this is called the Calving cycle. Now, you will understand that since we want water to split at the leaf level, there needs to be water there, and so that's why you need to water your plants. It needs to be well hydrated. And also, the Calving cycle takes place in this broth, which is called the stroma, which is the aqueous fluid of this part, and therefore you need water. If you have water stress, the Calving cycle will slow down. And this is of course well known for a long time, but in photosynthesis models that are used in hydrology and in climate models, I think Eduardo Dali, Ignacio and myself, and Tuzet independently in plant, cell, and environment, we were the first one just to put it in among the other limitations that are, of course, light, temperature, and CO2. So, to understand these impacts, let's just briefly see what the Calving cycle does. The Calving cycle is basically using ATP, energy formed by the light reactions, to capture carbon dioxide. There is an enzyme, which is Rubisco, which likes CO2, and therefore uses it and puts it on this cycle until, well, I'm really simplifying things, until some sugar is formed. It's called, the sugar has 3 carbon molecules, therefore the photosynthesis is called C3. Now there is a problem that Rubisco is an enzyme. It's a bit of an undecided lover. He likes both CO2 and oxygen. And if he gets CO2, he does carboxylase. But if there is oxygen, he will do oxygenase, which is the opposite of what he should do, and is not producing sugar then. And there is actually oxygen because that's the product of the first part of the photosynthesis. So let's go back to my story here. And, well, that was not a problem. At the beginning, there was not really much oxygen, and carbon was abundant. So this problem of Rubisco doing oxygenase was not really an issue. But at some point, oxygen became more abundant, and CO2 started dropping. And so it became important for plants to actually do something about it. And they came up with a very clever evolutionary strategy, which was to create a CO2 pump. And what they did is basically to say, look, we change our anatomy, and we split our leaf in 2 compartments. Now we have the mesophyll and the so-called bundle sheath cell. And in one, we do basically get rid of the Rubisco, and we do this, like, for example, with malate. Malate only likes CO2, and it captures it happily. And then we use a different compartment in which we don't let oxygen go. And there, we do exactly like before. We release CO2, and we send it to the Calving cycle. But there is no ambiguity now in having oxygen here, and so the efficiency is much increased. That's actually a very clever way to evolve on the same type of engine that we have, which is a chemical engine. And actually, this created much more water use efficient plants. Why is it called C4? I forgot to say that, because these are C4 carbon compounds. That's simply the name. Now, these plants are much more efficient. The we have, for example, the typical C4 plant is maize, corn, and this is dry biomass produced, and this is amount of evapotranspiration, and these are typical crops. So we have wheat, alfalfa, barley. You see they have all a typical slope here, and the outlier is the maize, which has much more dry biomass per evapotranspiration done. So much more efficient, and this is why we actually like, well, one of the reasons why we like growing corn, we like growing sugarcane, we like growing sunflower, which are all C4 plants. And this is an example of a review of different data in which you have evapotranspiration per growing season and production of corn. And you see really, once you have water to transpire, this is 12 and this is 4. So the production is really much higher. Now, of course, also notice the different behavior with wheat. Wheat is a C3 plant. It's more of a plateau. And this is more of a steep rising curve. We will see that reappearing in a moment. Yes?
But this is basically saying that you gain a lot of mass, right, from corn.
Yes.
But corn also is using a tremendous amount of water, right?
Yes.
So it's not that this is necessarily good.
So, yes, this is a very good point. In the next slide, I will, I will answer that. So it produces much more, but it also consumes much more. And actually, there is more to that, which is much more sensitive to water stress. So, yes, I completely agree. Before I want to make an analogy, which is what evolution did is to build around the C3 engine, chemical engine, adding a pump to increase the concentration of one of the elements that are used for the reaction, which is CO2. That's exactly what they did. And to me, it really reminds me of what we have done on our cars at some point in the evolution of the design, which is we use the same Otto cycle or diesel cycle, whatever, but the exhausts come out at a pressure that is high compared to the atmosphere, and we can use some of that to actually put a pump, which now, instead of pumping CO2 at a higher concentration, pumps oxygen, air at a higher concentration, and makes a combustion more efficient. It's a superficial analogy for us for now, but I'll elaborate on that later on. But then I come to the point of productivity and comparison of C3 and C4, and actually let me add immediately this. We did also a careful analysis of literature data. These are the parameters of the light reactions of photosynthesis. This is the type 2, the photosynthesis 2 that we mentioned before in our conversation. And this is instead, this is the compensation point of the Calvin cycle. If you don't know the parameters of the Farquhar model of photosynthesis, will not tell you much. But the bottom line is that C3 and C4, this is leaf water potential. So this is well-watered plants, and this is stressed plants. And you see they are different. And if you really want to synthesize all of this into a synthetic diagram, it's exactly what the question was before, is that this is evaporation over potential. And for C3, C4 grow a lot, but they also consume a lot. And as soil moisture goes down, they also fill water. Think of this as corn, and think of this as soybean or wheat. Sensitivity to water stress much higher. And then now in this phase, which is semi-arid, then C3 would do more in terms of evapotranspiration and probably also productivity. So it's quite natural now, being interested in larger scale hydrology also, and land-atmosphere interaction, thinking, look, we go to tropics, let's take, for example, Northeast Brazil here, and we had an Atlantic forest, which is basically C3 trees, and we are changing huge areas, and actually that has happened, with sugarcane, which is C4. So basically switching between the two evapotranspiration type of curves. At high soil moisture, lower at low soil moisture with C4. And we are doing the same with corn in many other parts of the world. And we are doing maybe slightly less, but still intensively also with another C4 plant. So the question is, there may be some feedback. There may be some impact on the local hydrology, exactly like I was mentioning before as a hypothesis, maybe increasing precipitation or something. So how am I doing with time? Halfway? Okay, so to study how plants affect local meteorology, I tend to choose minimalist approach, to use models that are simple, that I can control in terms of feedbacks and the way the processes speak to each other. And for this type of analysis, what I've chosen with my collaborators and my group is basically to just do a simple mixed layer model of the atmospheric boundary layer, which means that I consider turbulence strong enough to basically create a well-mixed atmospheric boundary layer profile. On top of it, there will be a free atmosphere, and between them, there is just a jump. Now, the jump is not, of course, a step change, but it's good enough for me. The key thing to realize here is that this well-mixed layer, which has also a logarithmic profile, grows during the day, as in the morning the soil hits the surface, the sun hits the surface. There is energy input that creates mixing, convection, and this height of the boundary layer, which I will call H later, is not the depth of the soil anymore. This grows during the day. And at some point, it may actually reach the so-called lifting condensation level, which is the point at which if you lift adiabatically some air, humid air that you take at the surface and you lift it up, at that point it will start condensing because it expands adiabatically, it cools, and it will condense. So that's the point. If you look at the clouds in an afternoon, sometimes in the summer you see a flat level in the clouds, and then you see really the flat and then the cloud like this. So that is basically the lifting condensation level, okay? So to have convective precipitation in the summer, which means thunderstorm, you need to have crossing of the lifting condensation level, but many times you see these clouds, it doesn't rain. The other condition is that the convective available potential energy is large enough, and there is a magic number that I borrowed from some colleagues in meteorology, which tell me more or less it needs to be 400 joules per square meter. The units here don't matter. I skip in the interest of time. So we can imagine very simplistically that our potential temperature, which is a mixture of pressure and temperature, so to speak, which remains constant if you do adiabatic mixing. That's why it remains constant, although the real temperature goes down. In the atmospheric boundary layer is constant. There is the capping inversion, and then typically above there is the free atmosphere, which in terms of potential temperature is warmer, just because you add the pressure. You take into account the reduction of pressure. So this will be my profile of potential temperature. Remember that these lines here grows during the day. And in terms of moisture, this is specific humidity, which means grams of water in the air per grams of air. Has a well-mixed profile also, a step change, and then typically the free atmosphere is drier, so you have a profile like this. This is my simplification, but it allows me to really get 4, which then I will reduce to 2, main quantity that describe the whole free atmospheric synoptic condition, which is this slope, the intercept, where is that? Here, QF naught, and gamma theta, which is the temperature lapse rate, and its intercept. So this will be my simplifications. Now, actually it's a simplification, but if I speak to Eli and his colleagues who do large eddy simulation, actually of course, they do lots of much more detailed turbulence, but if you just took the mean profiles, it's actually not far from my idealization. You see a relatively flat profile. Well, the change is not a step change, but we can actually show that a linear connection here would not change much my story. So I'll stick to my simplified profiles. So based on that, I'll just write the equations for the different layers that I had drawn before. And what basically we have is that at the surface pumps energy into the boundary layer and makes it grow. And the energy is in two forms. So we receive net radiation from the sun into the surface, and this, depending on the moisture, is split into sensible heat and latent heat, or evaporation and turbulent heat transfer. Now, if the soil is moist, this will be big, and this tends to be small. If the soil is dry, this will tend to be bigger, and this will be smaller. These fluxes are actually driven by both this one and this one, are driven by turbulent fluctuations, which are hidden into the atmospheric conductance. Now, let's leave it like that, but it's driven by turbulence, and by the gradients between the surface and the boundary layer of specific humidity and temperature. There is another thing that you see here, that not the sensible heat, but the latent heat has stomatal conductance. Now, the stomatal conductance is this control by the plants that open and close their stomachs to avoid transpiration when they cannot do photosynthesis. So if the soil is dry, this will go down because plants control that, try to control that. So this is where we will see the plants acting. Okay, this is the energy input, and these are the conservation equations in the mixed layer, which are the usual. This is the first law of thermodynamics. Amount of energy stored in the mixed layer is input of sensible heat from the surface, and this is entrainment from the free atmosphere. You see our parameters. This is theta f, is the profile of the free atmosphere, the linear one. As the dt, which is positive, grows, it entrains energy from the free atmosphere. The same for water. The rate of change of the amount of water in the mixed layer is input of water by evapotranspiration plus entrainment. And then there is an extra closure hypothesis that people commonly do, which is basically just to say how turbulence behaves in the capping inversion. So I'll skip the details on that. If you have questions, we can discuss later. So I have now, this is a closed system of equations. I presented it a little bit fast, but believe me, it's closed, and it allows me to grow the boundary layer depending on the surface conditions and the input of energy. Now, I also want to compute my CAPE, convective available potential energy. So I'll give you just very quickly the idea of how we compute it, which is, so if this is the profile at a certain time, so we have the ABL, and then the capping inversion, and then the free atmosphere. Now, to compute the convective available potential energy, which is this area here, we do this. We imagine that the turbulence inside the ABL is strong enough that it can behave like an adiabatic lifting. So we take a parcel from here, we lift it adiabatically, meaning with no heat exchange, and it's basically just going up at lower pressure, so it expands, and therefore it cools down adiabatically. As it cools down, here nothing happens, but at some point, the cooling is strong enough that it determines condensation. So that's the lifting condensation level. That's how we compute it in time, how it changes. And above that, the more you go up, the more expansion there will be, the cooler the air will be, and the more condensation there will be. Now, once you condense water, you release the heat of condensation. And so actually the parcel of air that you have will be slightly warmer. This is called the moist adiabatic profile. So instead of just going with a dry adiabatic, which doesn't have the extra heat added by condensation, from this point on, you actually have slightly warmer particles. Now, This is still cooler with respect to the free atmosphere up to this point at which the situation is inverted, and you get extra heat by condensation such that the lifting, the adiabatically lifted particle is actually warm, and it tends to be buoyant. And this is where you start convection. And if this area is big enough, Cape will exceed the magic 400 figure, which to which I didn't put any units, I know. And it will start the deep convection. So that's the story. Let's simulate this relatively simple system, and we see that during the day, the H, which is black, grows, the lifting condensation level grows, but not enough. At some point, the two cross, formation of fur clouds. Here we have the level of free convection, and I didn't mention on top of that there is the level of neutral buoyancy, which is where the situation is inverted, but that's not important. Now, these are different soil moisture, and you see that by changing the condition of the soil, we change the timing at which this happens. And here instead we have, well, something which is nothing, forget about the blue line, we're in a hurry. And just focus on the black lines. This is Cape. Okay, these are finally the units, okay? Joules per kilogram. It grows during the day, but of course, depending on the moisture that you have, it may grow more or less. And only in the last case here, it actually exceeds the 400. And it does it even before the crossing of the LCL. Now, where is rainfall going to happen? Well, most likely here, because you have crossing and Cape large enough, but not at this time because, well, I need actually to mention the blue line, which is this area. This is the convective inhibition, which is basically air that is cold enough, even if you lift it up to here, it's cold, and it will actually tend to sink down instead of going up. So you need to pass that, and you do that only when the LCL reaches, the H reaches the lifting condensation, the level of free convection. Okay, so the story is soil moisture really has an impact on those processes, and this is for some atmospheric conditions. Now, if we change atmospheric conditions, this is just before we move on, an example of real data in the Duke forest with Jenny Wang and Gabi Katul, students and colleagues at Duke. Well, this is a case in which both conditions, Cape and LCL crossed, and Cape was large enough, and sure enough, a little bit after we had rainfall. So the fact is that, and I think we were discussing this with Jim before, there is this dual aspect of both crossing the lifting condensation level and at the same time having enough convective available potential energy. And the situation is actually complicated by the fact that the free atmosphere can have different parameters. So these are almost like the whole parameter space. This is the lapse rate for moisture. This is the lapse rate for temperature in the free atmosphere. And this is a dry, wet atmosphere. This is a stable, unstable atmosphere. And the colors here are the soil moistures at which it is more likely to have convective precipitation, which we determined in this way. Now, I'm afraid that the colors have disappeared, but I'm not sure you can see a yellowish. But basically what we have here is that if I change soil moisture, this is A, so it corresponds to this point. So for this case, I actually have a dry soil advantage, meaning that this is the time at which, at the end of, well, at some point in the day, I cross the lifting condensation level. And this is the Cape at that time corresponding to that soil moisture for those parameters of the free atmosphere. And you see that if the soil is very wet, you have lots of cape, but you don't cross the LCL. The crossing happens, well, not before sunset, when there is then not enough energy to drive the ABL. So most likely, convection will be around here with relatively low soil moisture and not very strong precipitation because of low energy. But the situation is inverted if you go to point C here. Where is that? Here, in which actually the crossing happens, and it happens at all soil moisture, and therefore probably the most intense precipitation will be the one that has 600 cape, right? So that provides an overview of how we can have feedback from the land, from the surface to the atmosphere. Now we can ask the question that I prepared before by having the C3 and C4 differences, and now we can play with how the two evolves because of the different way they pump water into the atmosphere. And I mean, the differences are not huge, otherwise we would really be in troubles, but still enough to change of half an hour. This is C4 and this is C3, all the rest being the same. And this is dry and this is wet. And okay, first of all, the situation is inverted, as we would have, what am I doing here? No, sorry. This is the time of crossing, and this is cape. So the situation is dry, C3, C4, and changed. And so both in time is and in amount of energy, as we would expect. There is potential for changing the surface conditions into modifying the convective precipitation. Now, over the long range, those may be big differences in terms of hydrologic processes and biogeochemistry. Now, the last piece is, well, I have not forgotten that there is a third type of photosynthesis, which is probably the more interesting one, the one of these guys, the one that really know how to save water. Cacti, prickly pear, which is actually beginning to be used a lot for feed for cows and for even possibly for biofuel, but that's not sure. Pineapple, for example, orchids, all the epiphytes, aerial plants, they have to save water. Although they are in tropical wet areas, they take water from the air, so very little. And so basically what these plants do is something extremely interesting. They have split their process in day and night through a circadian rhythm. And they do the usual calving cycle during the day, but they do it with closed stomates, so they don't lose water. And they, at night, when everything is cool and humid and you can open your stomates without losing too much water, but of course you don't have light, they basically only uptake CO2, and they store it as malic acid in this vacuole. And by doing that, they basically save lots of water, and they can do much better than all the other plants. And let me continue in my analogy. It's basically what we did with the hybrid cars. No? You have a regular engine, again, Otto or diesel cycle, and when we don't use it, or when we brake, we go downhill, we store that extra energy, and that allows us to be much more efficient. So actually the analogy can be pushed a little bit further. It's if you think about it, it becomes quite interesting. First of all, there is the same evolutionary sequence. C3, C4, CAM, regular car, turbo, hybrid. Now, C4 is good at low CO2. The turbocharger is good at low oxygen. Now, for example, high elevations, airplanes or mountains. CAM has problems like the hybrid does, because of storage limits and costs. And the other thing that you discover, and again, I apologize, but this has disappeared, but these are data from CAM plants that shows that if the night, let me just give you the gist, if the night and the day are too similar, there is really no purpose in doing all this switch, because basically you lose water also at night. So if the night is very hot and very dry, you will not find chem photosynthesis. Cacti will not do well, which is very similar to what happens with cars. This is a relatively recent publication on that, which we found, which shows that for a hybrid to be more efficient, you have to have a city traffic type of scheme, braking and stopping. If you do highway, why should you bother charging your battery and redraining immediately? It's just a waste. And it's exactly the same for CAM. So actually, we're going to push a little bit more this analogy and see if something interesting comes out quantitatively. But if we have 5 more minutes, I would like to just quickly present how we modeled our CAM plans, because I think really we were the first one to put together the pieces, and we were quite excited about that, to really be able to compare with the other type of photosynthesis. And so the way it works is, our model has worked, and it took a while, but it's like, look, there is a Calvin cycle. Let's use the usual classic Farquhar model. It's driven by carbon dioxide at temperature. This is supposed to be leaf water potential and light. To that, we add this malic acid battery. We add the plant capacitance. Most of these plants are succulents, actually, so there will be just a storage capacity, like a capacitor. And then we have a nonlinear oscillator that does the circadian rhythm. So M is the storage of malic acid, which depends on the fluxes. I'm going to go very quickly here. And some variable which expresses it's called the gene expression of the CAM photosynthesis. It just tells you how permeable the membrane is, and it turns out to be, well, a nonlinear function of Z itself and M and leaf temperature, according to some data from the literature. The bottom line is that this creates this self-sustained cycle of storage and release. And the last piece, all stitched together consistently, is that the stomates open and close, as in previous models of photosynthesis, by basically demand-driven, meaning that they try to open and close, trying to keep constant the level of CI, which is the internal carbon concentration, to try to be as efficient as possible. And this is actually doing the job of opening a stomate automatically, because the Calvin cycle during the day draws down CI, and of course, if the plant is stressed, it will come from here, and the stomates will close because this will be low, and so they will try to increase it. And instead, at night, when there is demand from here, then automatically the stomates will happen. I mean, there are lots of details here. We're getting tired. I'll skip that. But if you're interested in the references are here. But the results were actually quite interesting. It reproduces well by itself without imposing any external, other than the day and night cycle, any closing or opening of the stomates, the phases of uptake of photosynthesis, recharging the malic acid, uptake of CO2 during the day. Of course, it draws down malic acid and so on. It's interesting because most of the data we found is for agave tequiliana. Now, they used the cactus to make tequila, so they have to study how it works. So clearly, there is potential for alcohol production here, no? Some ethanol. And or this is just to show you that it works well as you have a dry down. So this is 40 days. We try to kill a cactus, basically, by drying it in a computer. And at the beginning, it does well. And then, of course, it starts reducing photosynthesis. So this is the normal night, day, a little bit stressed, but I mean, just meaning reducing the photosynthesis. And interestingly enough, after a while, it just keeps everything closed, but the model even reproduces it's called the CAM idling, which means that the plants has anyhow to respire, and since everything is closed and there is CO2 because of respiration, the plants uses it. And so it basically uses its own waste and manages to push even further the production that, of course, other plants are not able to do. So we did some analysis of what happens if you perturb by periodic fluctuations slightly different from the ones of the internal clock, and there is a bit of before shutting down. This is a bifurcation diagram. It's periodic oscillation as it should, and when here like this is the amplitude of the temperature forcing. Before collapsing, it actually undergoes chaotic behavior before then giving up, and so we did some classical nonlinear dynamics of that behavior. But I want to conclude by just bringing everything together, if I can, and really put together the different type of photosynthesis. We have the C4, as we've seen. This is a dry down, so very efficient, very powerful at the beginning, quickly give up. C3, somewhat in between, and the strong cacti that these are two types of parameterization that resist much more. When you put them back into our stochastic soil moisture models that I mentioned at the beginning, remember I mentioned the frequency lambda. This is the last slide, penultimate. And you see that frequent rainfall, why should CAM do well? Of course, C4 are doing best. This is a total assimilation through many growing seasons. I skip the detail. This is like semi-arid. Now C3 is probably doing a little bit better until you get to very dry conditions in which now you switch to the, I mean, of course the assimilation is reduced, but the winner there is the CAM plant. And back to our analogy, if this is the ensemble average, the probabilistic ensemble average of assimilation through many growing seasons, as you change the rainfall frequency, you see advantage of CAM, advantage of C3, advantage of C4. And just to push a little bit more the analogy, this is gas prices in city driving conditions, and you have the advantage of conventional if the gas is cheap. I mean, this is price, again, no units, but it's like basically price per mile. And then, in this case, the hybrid is much better. And in this, for this parameterization, it's really not good to get a turbocharger. Okay. So in conclusion, it's a bit of a tour of many different appetizers that need to be more developed, I guess. But I think it's interesting because it shows some fascinating interactions between plants and their environment, how those dynamics may be important for impacting the dynamics of soil, the hydrologic cycle, and for us who are in an engineering department also and interested in sustainability, to put numbers and see really who does better in which conditions, and maybe optimizing crop production, biofuel, optimal use of water and soil resources. So thank you. This is for those who don't believe that cacti do very well in arid environments.
Questions? Please wait for the microphone so we can support your.
So, Amilcare, your model applications were mainly to look at differences between different plants. But if you, again, since you took us back in time, another interesting question I had is when plants started appearing, which you showed a big drop in CO2. So then not only are they affecting the daily cycle, but they're also affecting over the long term the initial conditions and the environmental lapse rates. So if you were to start applying this model from, let's say, soil, I mean, you run it with soil and then you add plants, and you would, I mean, are you thinking of any applications of that sort? Or if you run it, for example, just with soil, what difference it would make in terms of the atmosphere?
No, yeah, the atmospheric conditions is how all of that changes the free atmospheric condition, like the lapse rate. That's something that it's really worth studying. As I mentioned, this is very preliminary and recent. But yes, like making the land big enough such that you can have convective, you can have continental climate relatively isolated from the sea, in which the only source of moisture, which then stays and changes the free atmospheric conditions, then you would see these impacts, yes. And in these times, you had a full single continent, so it doesn't get more continental than that, right?
Are you using the steady state soil moisture?
Yes, yes. It's here.
In all the graphs, you know,
This,
when you
this
have
was done assuming a steady state.
steady state soil moisture.
Which
The amount of biomass is what we'll talk a little bit this afternoon, but in a different context. It's not playing a role here in any assumption of a steady state in the biomass.
we are not, if you want to think, yes, so you're perfectly right. This is like assuming this stochastic process keeps going forever, and plants are at the same, so they're not harvested, or they're actually harvested in such a way that
It could be interesting, just
yes.
as a comment, that I have no problem with the steady state of the soil moisture, because it's very quickly changing, especially in semi-arid areas, but in the biomass, it may not reach the steady state. I mean,
yes. I mean, the excuse that I have here is that these are meant to be crops.
okay.
So, but I completely agree with you. The feedbacks of growth of vegetation is.
No, thank you.
Thanks for the computer.
Sure. Amilcare. Exactly, my question is about this slide. So a lot of attention is paid to the basic changes in the rainfall. But you know, as climate changes, a lot of other variables like temperatures also change. Have you thought about how to look at the variabilities of other environmental controls other than rainfall
Not
in
at the
terms of
moment,
frequency
no.
on these plants?
Yeah, no, that's a good question. Not at the moment, although if you look at this, for example, this is amplitude of the temperature forcing on chem photosynthesis has a huge impact. So, yes, cacti, for example, may completely change their performance if the temperature changes. It's a very critical thing. So yes, very good question. We should do it.
I'm sorry if I missed it. I just wonder whether the model has a nitrogen limitation, or it just assume no limitation for nitrogen.
We assume nitrogen constant. So there is basically set nitrogen availability, but it would not be too difficult to do it by changing, where is probably, I should think about whether nitrogen has an impact here, but I may not have big impact. Certainly it has in the parameters of the Kelvin cycle as it is. So, but these have already been studied. That's why I think it is an advantage in this model to have to be built around something that is very well studied. So you can inherit all what has been done in terms of nitrogen availability from this study is really a classic, and it has been, so for example, the VC max parameter of the Farquhar model is known to depend on nitrogen availability. So it's worth having checking it.
Yeah, just quickly, the reason I asked it is because, say, if you use corn plants and soybean plants as an example, then they do have very different nitrogen requirements for the biomass growth, and I just wonder whether that would make a difference in terms of the different
Yes.
scenarios you have been doing.
Yes, definitely. Soybeans are nitrogen fixers, so they basically almost take care of their own fertilization, and you would see a third axis in the, we're doing reversal. So you would see a third axis here in terms of nitrogen, not just moisture availability, and these curves will probably be modulated by that.
As atmospheric CO2 rises and climate changes, in particular in terms of precipitation variability and mean temperature, do you expect like a particular type of photosynthesis to be favored?
That's a very tough question. So well, I mean, I can speculate. Now, CO2 increases, the need of C4 is less than before because there is more CO2 available. So I would, in terms of CO2 alone, definitely C3 would be favored. Now, but you're also, this is just pure CO2. Now, in terms of temperature, C4 like higher temperature. So in that case, now the C4 would be favored. If you have lots of variability in rainfall, it could be very dry. And now so my point is that there will be a readjustment of the niches, of the configurations in which each photosynthesis is favored. That would be my guess, but definitely worth understanding which direction. But there are lots of feedbacks in which it's hard to say which one will win. Does it make sense?
With that, why don't we give Amilcare a thank you again.